And again, welcome everyone to the second Forever Green update and project update and discussion. Walid agreed to share some of his work on oat and around heat stress, and although it's on oat, we don't necessarily consider oat one of the forever green crops, but certainly it is in terms of cropping systems. So Wally today is going to present his research and heat stress on oat and indicate how what they're learning may have impact across the full array of forever green crops and beyond. So Wally, why don't you do a little detailed update background on yourself, just to introduce yourself.
Sure.
And say a little bit about your program so people have a better understanding of who you are. If that's okay.
Sounds good. Are you seeing the screen?
I'm seeing the screen, and if it's okay, I'm going to record.
Not a problem.
Okay.
All right.
Okay.
All right, we're ready.
Thanks, George. Yeah, thanks for the introduction, Don. My name is Walid Sadok. For those who don't know me, I'm a crop ecophysiologist here at the Department of Agronomy and Plant Genetics. And my program basically looks at plants at the whole, the organismal level. And we try to find traits that improve plant adaptation to climate change triggered stresses. So we look at something we call atmospheric drought, heat stress, nighttime warming, freezing stress. And what we're trying to do is to find traits that are actionable for breeders so that they can develop varieties that are more resilient and work in various crops, oats being one of them, winter barley, soybean, wheat, Kernza as well. And it's the general idea across these crops. And here I wanted to focus on this project that was wrapped up this summer. And it's actually blueprint not only for Forever Green, but also for give a sense of my program, because it's highly collaborative, meaning that I collaborate in all of these projects with breeders and agronomists. So this project here focuses on oats, and the lead person is Jose Lopez, a plant physiologist who was a postdoc, and now he works in an energy company down in Texas. Did an amazing job, and in collaboration with Kevin's team. So would like first to acknowledge the different people that were involved in this work. Of course, Jose Lopez, a bunch of undergrad students who helped a lot with the field work, and a few members of my team. And of course, this could not have been done without collaboration with Kevin Smith Lab, who's a breeder, and his main crop is winter barley, but he works also on oats and silphium. I think he's in the attendance, or should be in attendance today. So if there are questions in the breeding angle, he'll probably take over. And his team was tremendously helpful in terms of the planting, the crop management, and also harvesting and quality traits as well. And of course, funding sources. So this was funded by the MDA and the USDA as well. So oats, my understanding is that this crop was really a major crop in the US and Minnesota. There was even a person dedicated to doing research here. I think it was a USDA person. And historically, the upper Midwest has been a major producing region, and it represents 66% of the US production. And Minnesota, as you can see on this map, is a major contributor as well. And perhaps you've been hearing the drumbeat of kind of the effort on kind of growing more oat in the state. And this is because of the increase in market demand fueled by companies such as General Mills and PepsiCo, coupled with an interest in these nutritional health values of oat seed. So they are rich in antioxidants, growth proteins, which are very healthy, and beta glucans, which are considered by the FDA as beneficial for human health. There's a claim there about its benefits in terms of lowering cholesterol and improving heart health. And of course, you know, we have this traditional landscape of corn, soybeans, sometimes wheat rotation. Adding oats is beneficial from environmental perspective because it diversifies cropping systems, it disrupts pest cycle, etc. So the problem with oats, and perhaps some of you from like the older generations know this, it has been obviously used to cover large acreage. And then starting from the 60s, there was this steep decline in acreage in the United States. And there is some literature trying to explain this. And basically there are three main explanations provided by economists. Obviously, the first one is economical, driven by government incentives for farm specialization so that farmers move away from multi-purpose farms, daily livestock and crop, to more specialized ones using cultivating soybean and corn. And on top of that, there's obviously mechanization. Much of the oat that used to be produced was also consumed by animals, especially horses, and then these horses got replaced by machinery. That's basically unavoidable. That's technological progress that reduces the demand. And through surveys, it appears that farmers started to kind of look away and look at other crops because of they observed a decrease in yield potential, largely associated with a decrease in resistance to rust. So there's this environment, and first indication that the environment here in the form of abiotic stress played a role in the farmer getting away from oats. But the other thing that relatively fewer people talk about is that it is likely that climate change has been operating as like this invisible hand in this decline. So during this window here, where oat has decreased, as you can see here, we know that, and these are statistically significant associations related climate pattern with yield at the global level. We know that during this window, yield crops took a hit, especially for comparable crops such as wheat, as you can see here. And most of the penalties were more severe in temperate regions, Minnesota being one of them. So there's there's this effect. Now, when it comes to climate change, it's a relatively vague word, and there are many abiotic stresses operating at the same time. So I'm going to take a quick step back here to illustrate rapidly three main stressors, and there are many others, but they vary depending on the crop cycle, of course. So there's an increase in atmospheric dryness. This is a paper we published recently in my group. This is not a topic that is addressed enough in my view, but suffice to say that the fact that the atmosphere is getting dry, we don't know why, by the way, is actually diminishing yield potential, even in reasonably well-watered regions such as Minnesota, for instance. So that's a threat. The increase in nighttime warming, we wrote a piece on that recently. It's a major problem because the actual global warming is occurring much faster during the nighttime relative to the daytime at a rate that is 1.4 faster, actually. And that's one of those components, and that we're looking at at the stress in my program. And of course, this phenomenon of heat stress or heat waves, which actually has major impacts on crops, even in well-watered conditions. So this is one of those take-home messages I will come back to several times in this presentation about the importance of high temperature stress, even in temperate regions such as Minnesota. And there is evidence for this. So there are a couple of studies conducted here in Minnesota. So there are two publications on the same theme, published in 2011 and 14. The references are indicated here. In both cases, the work is led by Kling et al. And with contribution from Joachim Wiersma, who's an economist in our department. And basically what they looked at the environmental conditions over a period of 14 years and looked at the yield and did multiple regression analysis and to see what seasons have the most impact, explain most of the yield variability, and what are the seasons that actually are negatively impacted yield. And what is very clear in their data set is that warm maximum temperatures later in the season, typically in the spring and the summer, are associated with decreased oat yield. So this was one of the very first studies in environments such as Minnesota, and here obviously the North Dakota, and other study. The second study here, they even did a trial in Canada as well, and the signal is still there. So that oat, which is a cool season grass, is actually reacting negatively to increase in temperature. And this is consistent with what we know about the physiology of oat. So the crop grows optimally in, at least the kinds of oats that are grown here in the US, and Minnesota specifically. They grow optimally at 21 degrees Celsius and start decreasing their growth at the temperature higher than 28 degrees. So it's quite low temperature here. Much more recently, and based on a much larger data set, and in this case in Finland, which is comparable in some aspects climate-wise to Minnesota, in this case, the data set covered 42 years, and you can see here the title of the paper that was published recently in Feedback Research. So I did this, I extracted this from the abstract. And here their message is that they did the same type of analysis as I mentioned earlier on barley and oats. And their conclusion is that temperatures above 28 degrees during the time frame of emphasis decreased the yields of both crops, with oat yields decreasing more than those of barley. And the conclusion is that this calls for introduction of new heat-tolerant varieties of both crops, but especially for oats in the warmer future climate. And mind you, this is Finland. So, and here's the second idea I would like to inject. When we talk about stresses like heat stress and drought stress, you don't necessarily want to imagine like a Mad Max kind of hellscape where everything is super dry, etc. You just need temperatures that are slightly beyond the physiological optimum of the crop. So I'm from North Africa. When people here say, "Oh, it's very hot," for me it's like warm day. And because physiologically I'm more adapted to those high temperatures, but for locals, they react to that physiologically, they respond negatively, they are uncomfortable with that. And for the crop, it's the same phenomenon. So what is really important to know about crops and how they respond to high temperature stress is that the effect of heat stress is not linear on crops. And one of those key figures that illustrate this very nicely is presented here. This is from a review paper by Prasad et al. Published in 2017. So here on both panels, you have florid fertility, which is an indicator basically of yield. And in here, the effect of a 5-day heat stress treatment where the day and nighttime temperatures were 36 and 26 degrees Celsius. And you can see that if you apply it after enthesis, this treatment for sorghum and wheat, I did it for several crops, you don't have any effect on fertility and minimal effect on yield. But of course, as you can see here, if this happens during enthesis and even before enthesis, you incur this more severe yield penalties. So this is another important idea, is that the sensitivity is window dependent. It's phenology dependent. And the literature is quite convergent on the fact that there are two kinds of meta mechanisms or groups of mechanisms that operate to explain these declines. So that the first one is when it happens, this high temperature stress, when it happens during enthesis, the opening of the flower exposes very sensitive external organs to high temperature stress, which can prevent proper anther dehiscence, pollen germination, or stigma receptivity in response to heat stress. And as you can also see here, is that when you apply the stress slightly before enthesis, you can also incur major yield penalties, and that's due to the heat stress effect on male and female gametogenesis when the gametes are being developed. So that's kind of a synthesis of what the literature says about the impacts. And as physiologists working with breeders in translational context, we need to know those mechanisms so that we can extract traits that could help for screening processes. Now, I talked about this in the context of sink limitation. So the sink organ here is obviously the flower, which can become the seed. It's a sink because it receives carbohydrates. And these carbohydrates are fixed by a leaf-through process we call photosynthesis. And also we know that high temperature stress can also negatively impact the capacity of the crop for capturing CO2. So this is an image to illustrate that these are those small machines inside of the leaf cells that we call chloroplasts that do the magic of transforming light energy into chemical energy, into sugars. So if you increase the temperature by the same to those kinds of temperatures here, you see this kind of denaturation of the membranes, and basically, technically, that leads to what we call photoinhibition and reduction in a photosynthetic efficiency and capacity. So that in that way, you can reduce also the source of carbon that is important for you. So based on these ideas, the goal of the project was to breed explicitly for enhanced productivity of Minnesota oat under high temperature stress. And here, the step was first to identify mechanisms that are relevant for wheat for high temperature stress tolerance. And based on that, devise a translational approach to identify traits that are amenable to high throughput phenotyping, which is not necessarily an easy task because there's so many things in theory that you want to look at, but you need to really be focused on the key traits. And this is what the presentation is going to be about. And obviously, after you identify these traits, you develop, you screen mapping populations or breeding populations to identify tolerance genes that will be included in the breeding pipeline. So for the time's sake, and also full disclosure, we need to fund this. So that's going to be our next step. But we have promising evidence indicating that we can do the third step. I'm going to focus on these two important phases here. Okay, so in terms of the genetic material used here, we had first group of lines, which we call the MN oat founder panel. And here the idea, and this is a classical experiment when looking at adaptation to heat stress. So we grow this panel in multiple locations and to capture large gradients of environment. And according to two modalities, so we have a normal planting date and then a delayed planting, which occurs 2 to 3 weeks later. And the idea here is to maximize the likelihood of exposing the reproductive window to higher than usual temperatures when you delay the planting. In addition to that, and we did this in St. Paul campus here, we selected what we call a core panel, which maximizes genetic diversity. And these are 30 lines that come from various breeding programs here. These are their names. The stars represent the locations of these different breeding programs. And such that they flower in a very, very tight window of 1 to 2 days, if I'm not mistaken. And that's very critical because we don't want to play with the flowering time because you can have heat escape mechanisms and can have the sense that you have high temperature stress tolerance, which is just only due to phenology. And we want to remove that covariate from our analysis. And in this case specifically, the experimental design called for applying what we call the heat tents, which is something I'm going to develop more detail. So the approach here under the heat tent experiment is to target those measurements that capture carbon source and carbon sink traits and yield outcomes. So we measure gas exchange, transpiration, stomatic conductance, photosynthesis. We measure leaf anatomy, canopy temperature. In terms of the carbon sink, we measure pollen viability, fluoride fertility, and other traits that, for the sake of time, I'm not going to dwell on too much. And in terms of yield and yield component traits, obviously, we measure yield and its component and quality, such as protein, oil, beta-glucans. And in terms of the heat tent treatment, this is how it looks like. So we have those single row plots that are covered by this structure here in the same developmental window in both years between booting and heading, as you can see here in this multi-panel figure that represents the environmental conditions during the field trials. So here on the left-hand side, here in the panels A, B, you have the temperature, C, D, you have precipitation, and E, F, you have a vapor pressure deficit, which is a measure of atmospheric dryness. So you can see here that the window developmentally took place at the same stage, although it was smaller in 2019 because it was a slightly hotter year. But this is how it looks like. This is Singer, Reina, technician in my lab at the time, and this is Daniel, a grad student, doing measurements of gas exchange. So another thing that is really worth mentioning, because I presume that you might ask those questions about the effect of this plastic film here. So basically, obviously, you noted that we're doing a artificial greenhouse effect. So you can see that in the environmental data within that window here for each year. So on again, this is the same kind of structure. In the first two panels, you have temperature. Here you have VPD, atmospheric dryness, and this is light. I'm going to get to that later. So the outside plots, which are not covered by this heat and setting, are reflected by the blue line here. And the heat stress plots and actually temperature, environmental conditions are reflected by the orange lines here. So you see that we succeeded quite well in both years in actually imposing high temperature stress. But the other thing that is worth noting that we were able also to impose it only during the daytime, as you can see here, and not during the nighttime, because as I hinted to earlier in this introduction, the effect of nighttime warming is quite different from daytime warming, and we wanted to remove that from our environmental treatments. Obviously, there was an effect on atmospheric dryness due to this treatment, but not to the extent that it resulted in a buildup of water deficits, which is something that we wanted also to remove. And the data, as I will show later, is consistent with that, i.e. no interference with drought here. We only looked at heat. And in both years, there was a small extinction of 22% exactly due to this plastic film. But even under the heat tents, the light levels were way above the minimal, which is where gas exchange saturates, which is 500 micromole per square meter per second. So in terms of the results, I would like first to start by the outcomes from the delayed planting experiments. Sorry about that. Let's go back to the presentation. So, so what we did here is we compiled yield data coming from those different locations that we used, including previous ones as well. And as you can see here, there is a strong negative correlation between temperature, average temperature or maximal temperature, in the same kind of window 18 days before enthesis and yield. So the regression is quite kind of, the slope is significant, and the R-square is actually reasonably robust for such a wide array of locations. And we're looking at yield here. And so this for sure confirms that the increases in temperature for this full season grass before enthesis is driving yield declines. So the problem clearly is important in Minnesota. And if you move the window beyond enthesis, these correlations become much weaker, meaning that the slopes become much flatter, indicating that the susceptible window is flower development here in Minnesota rather than seed field. In other locations, it's the seed field that is that coincides, like other location other than Minnesota. For instance, in Australia, that's what they see. So again, in terms of keeping injecting ideas, this is also a very important idea. What happened, the relevant physiological window in one location in one environment type would not be the same in other environment types. So it's an important idea here for breeding. So the susceptible window is flower development rather than seed field, which happens to be exactly the experimental design that we follow in the heat tent experiment. So as I mentioned, we measured the heat, the yield responses and its components and response to the heat trend experiment. So I'm going to spend a couple of minutes here explaining the structures of the figures before delving into their significance. So here, each time the gray part of the figure represents the year 2018 and the non-shaded part represents the year 2019. And so here you have the number, the box and whiskers represent the variability in those variables. And the N here represent the number of experimental units. So what is really clear here is that when you look at the here highlighted in the orange box, is that heat stress consistently in both years declined yield, right? And it would be like my kids say, duh. But yeah, it's a field experiment, usually very complicated. So in both years, we had a repeatable outcome, which is good. Then when you look at the quality traits such as protein and oil, consistently, again, you have an increase in protein as a result of the heat stress treatment and a decline in oil. And this is something highly consistent with the literature, which shows that, you know, when you impose either drought or heat stress on cereals, you have an increase in protein, which is concomitant with a decline in oil. It's kind of robust negative correlation here. Looking at the beta glucans, in the first year we have a quite significant decline. In the second year we have somewhat of a decline, but which was non-statistically significant, which is to say we did not have a decline. So there is some inconsistency here. So that is to say that beta glucans are likely less susceptible to heat stress compared to other quality traits. And the heritability of the yield here was reasonably high. So as I mentioned, we looked at those kind of traits and physiological processes that are really important for yield determination and important also relative to the phenological window, which is reproductive development. So here, when it comes to floret fertility, it was, in our data set, the biggest response to heat stress. So there was a huge decline in fertility, 50 to 60%. Now, the real surprise for us was when you look at this panel B here, because the literature always says, well, it is the pollen that is most sensitive to heat stress, and in other crops, that's what happens, but not here. So the first year we have somewhat of a decline, which is statistically significant, much more robust than what happened in terms of floret fertility. The second year we have a non-significant effect of the heat stress, and if anything, you have a kind of increase in pollen fertility but it's statistically insignificant. So that is to say, the pollen fertility did not play a major role in explaining the yield decline in response to high temperature stress. At the same time, we had a decline in floret number, but both significant in both years, but not to the same extent as the decline we had for floret fertility. And all of these combined led to the decline in grain number, which explains, of course, the decline in yield. But the key message here is that probably something related to the female reproductive organ sensitivity to the heat stress explain this major decline in grain number as a result of the floret fertility, because the pollen itself was kind of fine. So if it's not the male gametophyte, it could be likely the female gametophyte, or maybe the sporophytic tissue as well. As I mentioned, we also had the hypothesis about the impact of high temperature stress or heat stress on this source limitation, which is represented here in this bound. So here we have photosynthetic rates, and consistently in both years, the increase in temperature induced at the heat tent reduced photosynthetic rate. But you can see it's not a huge decline. It's statistically significant, but it's not a huge decline. So here to conclude, what is really obvious in our data set is that heat stress on oats impacted more the sink size of the equation, namely the reproductive process sensitivity to high temperature stress. And very likely it is the female reproductive organs that were sensitive to the high temperature stress, resulting in those yield penalties. So that's kind of the mid-talk conclusion here. So the other thing that we discovered also is represented in this panel here. So here it is, the letter E represents transpiration rates. And you could see in both years, the exposing the plants to the high temperature stress increased actually transpiration much more strongly in the second year, which was warmer compared to the first year. And this, by the way, confirms that we don't have interference with soil moisture deficit during the heat and treatment, because if that were the case, you expect these boxes to be lower, because when you have soil moisture deficit, you have accumulation of stress hormones that basically shut down the stomates and reduce transpiration. So we thought about this as potentially maybe the plants where there's a mechanism here at play of canopy cooling. So we looked at canopy temperature depression relative to the surrounding air. So a positive value here indicates LTD, leaf temperature depression. So a positive value here indicates the canopy cooling. And indeed, actually the significance increase in both cases. When you make that transformation, you see clearly in both years that plants, the genotypes started cooling their canopies, basically spending water, because they had plenty of water, they were not experiencing drought, to cool themselves. So this to us was very interesting. So we looked at the data in more detail. And this is where it became really interesting. So we did a correlation matrix. So basically correlate variables with each other. So in our data set, we had yield and its component and a variety of other physiological traits. This is a relatively complicated figure. So I would like you to focus on the panel B here, and more specifically on these regions in orange here. So this is the yield. And these are the circles represent both the level of correlation and the direction. So they are blue, so it means the correlation is positive, and the bigger the circle indicates the darker the color indicates that the correlation is robust. So of course, yield is related to floret fertility, floret number, grain number, as we can see, but also, this is where it becomes really interesting, it's positively correlated with stomatal conductance, with transpiration, canopy, temperature, depression. So this tells us that the genotypes that, this is our hypothesis, that the genotypes that had highest yields under the heat and high heat stress treatment had the cooler canopies. And you can see here that the effect of photosynthesis here, there's a correlation, but not very strong. So here, I did not mention here that in the method, but we did some kind of sophisticated approach called physiological path modeling to kind of statistically correlate, not statistically, mechanistically relate these things to each other. And you can see here that the decline in photosynthesis, which is represented by the letter A, is weakly explains yield, but the lower canopy temperature likely influenced in a way that still biologists don't know how exactly. We have our hypothesis, floret fertility. So when you cool the canopy, you improve floret fertility, which improves the grain number, which improves yield. So that's our hypothesis of that point. And this is where it comes to, after we dug deep into the mechanisms, now we put the reader's hat and try to translate them into traits that are helpful for high throughput screening, which is very important for breeding purposes. So to develop a method that will allow us to do that, we selected, based on the 2-year experiment during which we imposed the heat tents, 8 O genotypes that contrasted in their floret fertility response to heat stress. So basically, we selected genotype that had a lower slope here, and genotype that had a huge decline in their floret fertility in both years as you impose the heat stress. And then we decided to deploy what we call thermal high throughput phenotyping. So as the name implies, it's just looking, developing remote sensing approach to look at canopy temperature. We did here through, we deployed this approach through three steps. Obviously, what you need when you play with these ideas, you need a ground truthing measurement. So you need actually to be able to measure the temperature as sensed by the plant itself. And we do that through thermocouples, which are they're clamped on the leaf, and they have high accuracy, as you can see here, 0.2 Celsius. And these are connected to multiplexers and Campbell data loggers. And then, dependently on the day, so on flight days, meaning that when we have days, sunny days, of course, and with reasonably low wind speeds, lower than 20 miles per hour, we can use a drone on which we mounted a thermal camera to take thermal images of our plots. And the accuracy here is fine. And since then we acquired another camera which has a higher accuracy and a drone with higher capacity that can fly in more windier days. And on non-flight days, meaning when you have like that wind limit, so when you have windy days, you can use this handheld forward-looking infrared camera. It has a higher accuracy relative to this camera here. And basically you go to the plots and you take your picture. This is the picture in RGB mode. This is the thermal image. Now, the name of the game is to compare these outputs from the drone and from the handheld camera to the ground truthing approach and see if these measurements are coherent, and then see what we have in terms of our hypothesis. So this was conducted, as I mentioned, by Jose. He did many things, and he was very keen on playing with the drone. This is Bishal, postdoc, who helped as well during that work. This is what our plots look like from above. And this is an example of given day presenting these plots in a color-coded fashion. So the warmer colors reflect higher temperature, and the cooler colors represent a cooler temperature. There's an entire pipeline developed by Jose here to extract the temperature information based on the median value of the pixels in terms of their temperatures. And there's a lot of cleaning and coding there. So in terms of comparing the thermocouple to the other sensors, just quickly here. So if you look at the thermocouples and the handheld thermal camera, you have a good agreement, and this is based only on four dates. So this was, we're happy with that. It's close to the 1-1 regression. So that means that this technique is actually quite effective. And actually it's much cheaper than using thermocouples because they're very difficult to install, and they're expensive because of the data logging needed, and that's very expensive. Then when you look at the relationship between the handheld camera and the drone mounted camera, you have also a reasonably strong relationship. It's not exactly on the 1-1 regression. And this is due to the nature of the beast, meaning the tools that we use. So we first, in this case, both measurements were done by the same person. So first the person flows, who was Jose. So you fly the drone, then you take the handheld images 30 minutes after the flight. And so those measurements of canopy temperature are very sensitive to wind speed, cloud cover, etc. So if you take measurements 30 minutes after that, you might have shifts, and also an additional 30 minutes was needed to collect images through the handheld device. So, but notwithstanding, you have a reasonable and strong agreement, actually, in my opinion, between measurements using both techniques. All right, so this was to validate the technique, and now the verdict is when you try to relate the thermal data gathered on those 8 selected lines and their performance during the heat tent experiment, right? So on the y-axis, we have the florid fertility during the 2 years of the heat tent experiment, and conducted during 2018 and 19. And on the x-axis, you have the drone canopy temperature measured through in 2020. And the nice thing here is that we found a very nice correlation between both variables. And here, what is basically this relationship is telling us is that the genotypes that were most weakly responsive to the heat stress, meaning that they maintained best their yields in response to heat stress, meaning here, for instance, this guy here that maintained fertility at 65%, had the lowest canopy temperature. So we have a robust relationship here. So in a nutshell, this confirms our hypothesis that canopy cooling is an important process for improving tolerance to high temperature stress for oat grown in Minnesota. And not only that, now we have a method that is high throughput friendly, which consists of flying a drone equipped with a thermal camera with an adequate image analysis pipeline to screen potentially hundreds, if not thousands of entries. So I have just 4 takeaways here. I hope I was able to convince you that heat stress is a major yield limiting factor for oat grown in Minnesota. It doesn't require excessive temperatures to reduce yield due to high sensitivity of the crop. Yield losses are largely due to floret fertility losses, which presumably we hypothesize that relates to the female gametophyte sensitivity. Canopy, maybe panicle cooling, appears to be a promising trait to mitigate yield losses, and now we have a clear pathway for remote sensing high throughput phenotyping protocol to identify the genetic markers associated with tolerance to high temperature stress. And we started applying this, and we're thinking about applying this to other forever green crops. So with that, thanks again for your attention, and please feel free to ask any questions.
Thank you, Walid, for a nice piece of work. And I think, like you're saying, I think it will have application to a wide range of crops, including the forever green crops that are currently under development. So before I ask questions, why don't we open it up for comments and questions from the group? So I'd encourage you just to open up your mic and ask your questions.
Very interesting talk, Walid. So one thing I wanted to ask about, you mentioned that the susceptible window in Minnesota was flower development, and I think you said Australia, it was seed fill. Do you have any idea what could be driving that difference?
Yeah, thanks, Anthony. It's mostly about when the stress happens, right? So in Australia, it's obviously hotter, so they have presumably genotypes that are more robustly tolerant to heat stress, but still there, the biggest waves or the most intense waves take place during seed fill. So they looked at that, and that's what they found. Here, it's just the environment, the environmental scenario. We call that envirotyping. We're putting together a piece with Kevin on that. How it is important not only to think about the phenotyping, you have to look at the envirotyping, because dependently on the environment, you might have different window for susceptibility, and therefore different mechanisms to look at, and therefore different types of traits.
Hi, Walid. I have a question.
Sure.
I'm an alfalfa breeder. You find the correlation between the yield decrease that associated with the heat tolerance. And as you may know, alfalfa yield in past 100 or more years, their yield not increase. Well, they not going down like oat, but they are like flat. So what's the main reason to make you think about your research found heat tolerance is the yield decrease, not other factors?
Yeah, that's a good point. So I hinted that, I mentioned that in the introduction. So there, obviously farmers adjust planting dates and things like that to kind of deal with the climate. But the biggest argument in favor of that are those yield environment associations. So basically what those groups in Finland, and by the way, they were using my understanding is that many Finnish varieties actually have common ancestors with Minnesotan varieties. So it's actually is which is the case in many papers about the effect of climate on crops. It's a robust multiple regression statistical analysis that show that there is a strong association between specific climate scenarios and yields. And you're right, in some crops you don't have that. So in their study, they showed that barley is also impacted, but to a lesser extent than wheat. So this goes in alignment with your point, which is it depends on the biology of the crop and also the way it's cultivated as well.
Yeah, but
Yeah.
alfalfa has a deep root and has a very high transpiration rate,
Yeah.
right?
Yeah. Yes, relate to that, heat and relate to drought tolerance. And they are probably on two sides of the one thing. And you also mentioned the drought tolerance. Will you plan to do any further study, put a combination of drought tolerance, heat tolerance to see how I know they affect each other for sure.
Yeah.
So which one is the main reason? Is the drought, we don't have enough water, or is the heat, as you mentioned, damage the flower, etc.
Yeah, that's an interesting idea. And obviously heat and droughts are the subject of many papers. And actually this is was the angle we took for the review paper. If I may share the screen again, I can show you here so you can see in detail the kind of the gymnastics that we went to. This one here.
Yeah.
So basically focuses on the question you raised. So the issue, the challenge with drought, if it co-occurs with heat, is that for cereal grains, the best strategy to withstand drought, meaning to maximize the chance of having higher yield and in water deficit conditions, is that to deploy a conservative approach. So basically your stomates need to be closed to save water, such that you bank enough water so that your seed fill phase, which is the most sensible to drought, is less affected. So it's a conservative approach. So if you have heat stress on top of that, and you're saving water, you cannot spend your water to cool yourself. You have a trade-off there. So that's the framework we operate in, and our hypothesis that if you are in places where you have deep soils with good soil moisture storage capabilities, and an environment where you have stochastic rain, which means that you don't have terminal water deficit, it is worth it to have genotypes that spend water, that throw it in the air to cool the canopy. If you're in an environment where you have drought, then you have to, the thing becomes more complicated. So you have to parse out new layers of mechanisms to kind of see what trade-offs are acceptable and what trade-offs are not.
Walid, do your lines that have the ability to cool the canopy have the largest root systems?
Obviously, we did not look at that.
I'm just saying, is that a possibility?
It is a possibility, but I would add to that is, so I did some work on wheat, specifically in the, from this perspective of interaction with drought. Sometimes you have, so differences in rooting depth do not necessarily imply differences in water capture. Because what really matters the most is the piping inside of the roots. So some root systems have different hydraulic conductances. And for wheat, we noticed that very strongly, which is at equal root depth, the most drought tolerant ones are the ones that are able to regulate the piping, how much water is going through them, either through regulating the xylem vessel size or the radial water flow through aquaporin. So rooting depth is the first step, but usually you have to go to the second step, which is to look at the hydraulic properties of the roots themselves. So it could be a possibility, but in our case, we had enough precipitation. We calculated that before we had the heat tent installed. So yeah, that worked well for us.
That would be a good, yeah, that will be good study for root structure, change your canopy type, then change your temperature, well, potentially. So
Yeah, I mean, I worked a lot on drought, and for sure these experiments are important, and you need to do them on location.
Yeah, but the idea that you're presenting, as I'm thinking about this, it may not be that it's drought. It may be, as you're describing, that the structure of the xylem or the root structure is such that as the temperature goes up, you don't have a high enough transpiration rate to cool the canopy, right?
Yeah, so that's a hypothesis. So the recommendations, that paper just advertised, is we ask people to look at that hypothesis.
Yeah.
Because we know that roots can act as sensors for temperature. We don't look at them that way. And what they can do is that they can fine tune, they accumulate aquaporins to maximize how much water is going through them. So the plants can regulate, the roots can regulate themselves. If they sense that the soil temperature is getting a little high, they can fine tune aquaporins to maximize water flow through the roots.
Is that mechanism published?
So we review the literature that point to that, but we hopefully in the future we can really present strong definitive evidence for that.
So you just put pieces together, right? In terms of that.
Now we had a couple of papers that really show that root systems respond dynamically to water availability and to temperature. So.
Wow. Cool.
thank you.
Thank you.
So question on heat stress and test weights for oats. So is that kind of inverse correlation well established? So it wasn't measured here. And then if you breed to bring yield along to deal with your future heat stress, does the test weights kind of come along with the breeding in that?
Yeah, that's a great question. So this opposite direction that we observe between yield and protein content, and between protein and oil and starch is widely observed in the literature, and it's the mechanisms are not fully clear. One possibility in the context of heat stress is that you have less starch. So in the seed, so in relative terms, the seed accumulates more protein, so that's why it increases. And you have less starch because the grain respires more because of the heat stress. Heat stress pushes the plant to respire more. But for sure, what we want to do, and when we screen, when we develop this at the high throughput level, is to look at those quality traits: the protein, the oil, the beta glucans, and see if we have genetic variability, strong genetic variability, and if we can detect genetic loci that will minimize the impact on heat stress on the quality. So for sure, it has an effect on test weight, likely on the test weight, on other technological traits. The goal is to help develop a program that minimizes those effects. I don't know if I answered properly your question, but I guess in a nutshell, we don't know yet, but we want to do it.
Yeah, it did. In our quest, to increase yields. I mean, that's always the trade-offs. Is if the other qualities that we look for, is it going to bring it along? Is there, going to be a detriment or benefit? I guess so. You answered it. Thanks.
I mean, beta glucans seem to be not impacted, which is good news, right? But this is based only on 2 years, maybe 3 years, or maybe more severe heat stress treatments would show us that there is a decline. It doesn't seem that all of those quality attributes respond the same way. So there will be some trade-offs there for sure.
Oh, Dan, can I ask another question?
Yes.
How you select the material? For example, as a breeder, we always screen large number of germplasm for study. You use the 2 or 8 germplasm to see the clear difference. But how you select those material for your contrast? And then if you know how you select those germplasm, I wonder among the old germplasm collection, whether they have germplasms, they're even sensitive to the temperature than what you use. Well, they have some non-sensitive materials for breeding purpose. And the long story short question is how you find those materials to start your research.
Okay, I have a short answer, but maybe Kevin can, I see his name, he can provide his input because he developed.
Sure. Sure. So Walid referred to this founders panel, which is like 260 lines or something like that. And when we restarted the oat breeding program, what we did is we looked at regional trial data for the whole Midwest over the past 10 years. And we selected lines that seemed to perform well across that region over that 10-year period to try to build a fairly broad base. But these were all, elite breeding lines. So they were lines that were pretty advanced in individual programs, but they came from Illinois, Wisconsin, North Dakota, South Dakota, Minnesota. There were a couple from Canada. So they were spread throughout the Midwest region in terms of breeding programs, but they were generally elite material. And then from that set, Walid selected a subset, this core set to work on.
Thank you. Thank you, Kevin.
Any additional comments or questions for Walid?
So Don, can I ask a question that's not directly related to this presentation, but you got experts on the screen here that can lead me in the right direction?
Go ahead,
Right.
Jeff.
All right, so this is related to a meeting on climate change I had earlier today, where we're coming up with scenarios about, and here's what applies to you guys. So if we were to replace an acre of corn with Walid and Kevin, small grain, Don and others, perennial grains, what kind of greenhouse gases, would we actually, reduce? So I don't know if I framed the question right. So that is a real question the state of Minnesota is, asking and trying to come up with scenarios. So I'm not looking for an answer, although appreciate your initial thoughts is like, send me in the right direction on helping me to understand the, the answer to that and the status of research on that. Thanks.
Yeah, I can take that one. And obviously everyone else is welcome as well. So my guess is what you're talking about is the carbon sequestration, right? Component.
Certainly carbon, but could be the nitrogen management as well. So it
Overall
could be
greenhouse
nitrous oxide.
Yeah.
gas.
So okay. So I'm gonna break that down into the management and the and kind of the biology component, right? So the management, if you apply parsimoniously nitrogen in very smart way, and you can reduce the greenhouse gas footprint of your cropping system. In terms of the carbon sequestration component of the equation, there is work currently so obviously a crop like Kernza, which has a profuse root system, so it captures a lot of atmospheric CO2 and dumps it in the roots. That's a carbon sequestration mechanism. We hypothesize that type of trait could be also expressed in annuals, although to a lower extent. So there are a big project, I'm not involved in those, but explicitly with the goal is to improve root phytomass in a way that would not generate a yield drag, such that you increase the carbon sequestration capability of the crop. So this is an area that is being investigated. So yeah, short answer, the carbon sequestration equation is being looked at. Perennials probably are better than annuals in that respect. And there's the management box, which is huge, right? So we have to deploy management techniques that are much more effective. And this is why precision agriculture those types of technologies are supposed to help with really optimizing input application.
Thank you. And I neglected to say we have an alfalfa expert too, so that comes into play here too, of course.
Yep.
So Don, can I say something? I know it's late.
Yeah, yes, but I'm going to be the last.
Okay. The Josh Gamble did publish a paper. He compared the carbon sequestration with plant alfalfa and the soy and the corn. And the conclusion is alfalfa probably is the most that sequester the carbon the most. So I believe that, and since I'm alfalfa breeder, I'm breeding for that. And I read the proposal this year, and that's my belief.
Well, Jeff, you probably know how I feel about it. It's a very open question. And if you look at most of the long-term trials, like the ones in Wisconsin, where you're probably at now, Jeff, basically, when you look at carbon sequestration, most of all of the disturbed systems, or I should say, all of the disturbed systems basically do not sequester carbon. They may reduce the loss of carbon. And based on most of the data that I've seen, the systems that do show carbon sequestration are the ones that are in fact perennials, the ones that are not disturbed over long periods of time. Most of my review in terms of cover crops, those types of things, I'm convinced if there's tillage associated with those, and certainly here in the upper Midwest, when you look at winter annual cover crops, the disturbance that goes into the system, most of the evidence suggests that you're not going to get a high level of carbon sequestration. I think the data suggests that you might, if you have high levels of biomass coming in from cover crops, you might be able to reduce carbon loss. But I think there's very little evidence that these systems, disturbed systems, are really positioned to sequester carbon. And there's evidence over the last 150 years, right? In our agricultural systems, over that period of time, we've lost 50% of our carbon, right? So I think there's going to have to be a dramatic change in the agricultural systems towards perennials that are on the landscape for extended period of times before we're really going to be able to sequester carbon. Now, greenhouse gas exchange, that's a different story, right? There may be ways of reducing greenhouse gases within these systems by managing nitrogen rates and things and tillage, but when it goes to that carbon sequestration, and that's where everyone is looking at now, I think there needs to be a real look at the real data to really lay out an appropriate picture for expectations of carbon sequestration with some of these systems that are being proposed, because the data suggests that the only time you sequester carbon are in those pasture systems, rotational grazing systems that are in place for many years, and the manure is returned directly back to those landscapes. Right. So right now with Kernza, I mean, it's now being investigated, as Walid has said, and I just heard a report today in the early stages of evaluating Kernza. It's still not convincing story that even Kernza is early on sequestering carbon, right? But the data is still out, right? So they're still gathering that evidence to get a real picture for greenhouse gas exchange and carbon sequestration in that system. All right.
Don, can I add something to, I'm sorry.
The question is, right, you go through and you have alfalfa in for 4 years. What happens to that carbon as you rotate to corn? Or with Kernza, what happens when you have Kernza in for 4 years and then you rotate to another crop? What happens to that carbon that has been placed in that system? Is it stable? Is it lost? I think there's still some key questions that have to be answered there.
The other thing I wanted to, just because you just triggered the thought, Don, good points. The climate change is kind of the mixing these things, because if you have warming, you increase respiration, and when you increase respiration, you lose that carbon again. We don't know how Kernza respond to that. We don't know how many other crops respond to that. So that's one problem. And the other problem, which is, you know, we cannot focus on everything, but it's like a meta problem, which is like beyond what we're doing, is maybe like agriculture, like biosystem engineering folks look at this. I looked at this during my first postdoc, which is life cycle analysis, meaning that you can perhaps imagine a crop that's really good in sequestering carbon, that have a good environmental footprint, like near zero, for instance. This is a theoretical case, but if the value chain, the industry, has a huge carbon footprint, it's kind of, you nullify that effect. So when you look at the system, we need a systems view. So each one does his part. There's the agriculture that needs to do its part, but also the transformation as well that needs to do its part. Otherwise, you might have ending with a situation where you actually deploy good crops, but you don't see the end result, because the industries are still being very consumptive and have a huge footprint. So that's another component that we cannot do. It's not in our expertise, but that's a fact.
Great. Thanks for entertaining my question, and great answers from all of you.
Well, you can, Jeff, you know as well as the rest of us, and it's a wide open question. We just don't have enough long-term studies to really say a lot, in my opinion. Others don't agree with me, but to really make concrete predictions as to carbon sequestration from some of these systems, there just needs, in my opinion, more investment to really do the detailed work, and it's expensive work to generate that information.
Yep. Thank you all.
Walid, I'm glad you're in our department. I'm glad that you're at the University of Minnesota. And as a former plant physiologist, I'm glad to see that plant physiology is coming in at a time and rebuilding these relationships between plant physiology and plant breeding. I think this is a great example of these relationships that, when I arrived in Minnesota in '74, these were the types of things that were going on. And then all of the physiologists retired and not replaced. So this, to me, is really, really exciting example of that potential long-term engagement of plant physiology in support of, or should I say, plant breeding in support of plant physiology.
Works both ways.
You guys know.
Thanks, Don. Appreciate it.
Yeah. So thanks again, Walid and Kevin and the rest of your team. So great presentation, and looking forward to looking at this research impacting the plant science programs here in Minnesota and across the country and around the world. So thanks again.
Thanks. Thanks, everyone.
Thank you.
All right. Bye-bye.